Okay, going to start the lecture. Okay, today we are going to discuss about the bacterial peptidoglycan biosynthesis. As you all know, peptidoglycan is a main component of the cell wall of the bacteria. Now let's see some video regarding the peptidoglycan biosynthesis. Okay.
Most bacterial cell walls contain peptidoglycan. This structure is essential for maintaining the structural integrity of the cell. Peptidoglycan is a structure that is unique to bacterial cells. This combination of essential and unique makes most bacteria.
Okay. The video which basically consists of how bacterial peptidoglycan biosynthesis works. Okay, I think slide is not able to transmit messages.
Most bacterial maintaining the structural integrity of the cell. Peptidoglycan. Most bacterial cell walls contain peptidoglycan. This structure is essential for maintaining the structural integrity of the cell. Peptidoglycan is a structure that is unique to bacterial cells. This combination of essential and unique makes peptidoglycan an excellent target for antibiotics. The precursor subunit of peptidoglycan, UDP-N acetylmuramic acid, is synthesized in the cytoplasm. A lactyl group is added to carbon number 3 of UDP-N acetylglucosamine to form UDP-N acetylmuramic acid. 5 amino acids are added sequentially to UDP-NAM to form a pentapeptide side chain. Special adding enzymes are used to add these amino acids so that tRNA and ribosomes are not involved. In the case of Escherichia coli, L-alanine is added first, then D-glutamic acid, then mesodiaminopomelic acid. Finally, the dipeptide, D-alanyl D-alanine, is added to form the pentapeptide side chain. The NAM pentapeptide is transferred from UDP to bactoprenol phosphate at the membrane surface. Then NAG is transferred from UDP-NAG to the bactoprenol NAM pentapeptide to form the final disaccharide pentapeptide precursor. The completed NAM-NAG precursor is transported across the membrane to its outer surface by the bactoprenol carrier lipid. The disaccharide pentapeptide is then transferred to the growing end of the peptidoglycan chain to lengthen it by 1 repeat unit. As the bactoprenol pyrophosphate returns to the inside of the cell membrane, one phosphate is removed by a pyrophosphatase, leaving a bactoprenol phosphate to accept another NAM pentapeptide. Finally, the peptide side chains are linked together by transpeptidation reactions, forming peptide bonds. In E. coli, the free amino group of diaminopomelic acid is linked to the carboxyl group of the subterminal D-alanine, releasing the terminal D-alanine. Other enzymes remove the terminal D-alanine from side chains that are not cross-linked. Most bacterial.
Okay, now moving forward with the gram-positive and the gram-negative cell walls. Gram-positive and gram-negative cell walls. Okay, the gram-positive and gram negative cell walls, how they work actually. Okay, what is the composition of gram negative and gram positive cell walls. Okay, so how are the synthesis of peptidoglycan occurs? Okay, so we'll move forward. I think there is some technical error right now, which I'm trying to figure out. I'm not able to. Okay. Uh,
Most bacterial integrity of the cell. Excellent target for antibiotics. Most bacterial
okay, gram positive and gram negative cell walls. As you already know, gram positive cell wall contains peptidoglycan in more content as compared to the gram negative cell wall. Okay, so there is a difference in the composition of gram positive as well as gram negative cell wall. The diagram given here is an gram positive envelope is from Bacillus laciniformis, which is an gram positive one, and the gram negative one is of Aquaspirillum serpens, which is given here. And the peptidoglycan layer or the murin layer, okay, this one is a peptidoglycan and murin layer, okay, and this one is an outer membrane given here, okay, then comes a plasma membrane, okay, then comes a plasma membrane, okay, and here given is in gram negative cell wall, where you can clearly see the amount of peptidoglycan is very low in the gram negative bacteria as compared to gram positive bacteria, which has a higher peptidoglycan content. So starting with the process of the biosynthesis of peptidoglycan, it starts with the synthesis of UDP NAG and UDP-NAM. Okay, and what do you mean by UDP? It is uridyl phosphate. It is uridyl phosphate. And what is NAG? N-acetyl glutamic acid, and NAM, N-acetyl muramic acid. Okay, so let us discuss first about the UDP-NAG. Okay, it is formed from the fructose 6 phosphate. It is formed from the fructose 6 phosphate and converted into glucosamine 1 phosphate, and in the presence of acetyl CoA, which will be converted into CoA, and N-acetyl glucosamine 1 phosphate will be formed, and finally with the help of UTP, phosphate will be released and UDP NAG will be formed. Okay, UDP NAG will be formed. Okay, this one is a short overview of the process of the formation of UDP-NAG, which is uridyl diphosphate and acetyl glucosamine. And now moving forward with the UDP-NAG. From UDP-NAG, in the presence of NADPH and PEP means phosphoenol pyruvate, okay, UDP-NAM is formed. Okay, UDP-NAM is formed. What is UDP-NAM? Uridyl phosphate and acetyl muramic acid. Okay, and after the conversion of the UDP-NAM, okay, sequentially amino acids are added. Okay, sequentially one by one amino acids are added to the UDP-NAM, okay, forming a pentapeptide. Okay, so there is addition of 5 amino acids sequentially. There, which amino acid will be added may vary according to the type of bacteria. Okay, there are some different amino acids with different bacteria in a general. And next one is a important carrier molecule, we can say. Okay, a bractoprenol NAM, bactoprenol NAM. Okay, this one is a structure of the bactoprenol. This one is a structure of a bactoprenol, and it's an 55 carbon isoprene derived alcohol that is connected to the N-acetylmuramic acid by the pyrophosphate, Okay, now moving forward with the process of peptidoglycan cell wall synthesis. Okay, as in the first part of the process, we have already seen that UDP derivatives are made. Okay, which are they? UDP-NAM and UDP-NAG. Okay, in the second step, 5 amino acids are added, forming NAM pentapeptide, Okay, now comes the third one. Okay, now comes the third one. NM pentapeptide is transferred to the bactoprenol phosphate, generating lipid 1, and they are joined by the pyrophosphate bond. Okay, you can clearly see the pyrophosphate bond. Okay, bactoprenol and NM pentapeptide are joined by the pyrophosphate bond. Okay, and in the next steps, UDP-NAG will be converted into UDP. Okay, will be converted into UDP. Okay, and this serves as an interbridge. Okay, and glycyl tRNA molecules, but not in chromosomes. Interbridge formation occurs at the cytoplasmic side of the membrane. It occurs at the cytoplasmic side of the membrane. Okay, now in the lipid 2, okay, NAG from here will be converted into, will be joining the NM. Okay, will be joining a NM. Okay, which is joined with the bactoprenol phosphate with the help of pyrophosphate bonds, and this whole molecule with the help of bactoprenol, okay, where lipid 2 is flipped across the membrane by the enzyme flippase. Okay, this bactoprenol complex with NM, NAG as well as pentapeptide will be passed on to the other side of the membrane. Okay, other side of the membrane with the help of the enzyme flippase, Okay, and now moving further with the process, the NAG NM pentapeptide is attached to the growing chain of the nascent peptidoglycan chain that is held at the membrane by a bactoprenol membrane, and this increases chain length by 1 repeat unit. Okay, as you can see clearly, let us assume this is a chain of peptidoglycan, okay, which is being formed. Now what will happen? This chain will get attached, okay, will get attached to the system, and bactoprenol will be freed again. Okay, bactoprenol phosphate will be freed again, and NM NAG pentapeptide will join the peptidoglycan layer, forming a new layer of the peptidoglycan. Okay, and after bactoprenol is released, okay, after the bactoprenol is released, this bactoprenol now moves back again to the cytoplasm, and loses one phosphate, becoming a bactoprenol phosphate now, which can be again reused for the continuation of new formation of peptidoglycan biosynthesis. And the peptide crosslinks between peptidoglycan chains are formed by the transpeptidation. Now further process, okay, how are they joined? Okay, once the chain is added or a unit of NM NG pentapeptide is added, how are they joined? They are joined by the transpeptidation process. Okay, I am repeating the whole process again. Okay, process starts with the formation of derivatives of NM and NAG, which are N-acetylmuramic acid and N-acetyl glucosamine, where UDP is a uridyl phosphate or okay, and upon the formation of UDP and NAM, okay, 5 amino acids are added sequentially. 5 amino acids are added sequentially, and moving further with the process, in the presence of bacteropenol in the cytoplasm side of the membrane, okay, UDP will be converted into UMP, and bacteropenol and NAM pentapeptide will be formed, and they are joined by the pyrophosphate bond. They are joined by the pyrophosphate bond. And moving further with the process, UDP-NAG now will be added, and UDP will be again released, and here NAM will join the bacteropenol NAM pentameptide complex. Okay, now this bacteropenol complex will be further transferred to the another side of the membrane with the help of the enzyme flippase, and this unit of NAM-NAG pentapeptide will be added to the peptidoglycan chain, forming a new chain, that is new chain will be formed, and bacteropenol will be again freed. Okay, bacteropenol will be again freed in the form of bacteropenol diphosphate, and again bacteropenol for the further use will be again transported to the another side of the membrane, where one phosphate bond will be removed and bacteropenol phosphate will be formed. Okay, so further in the process, this peptide cross-links chains and peptidoglycan chains are formed by the transpeptidation process. Okay, this is whole of a process of the biosynthesis of the peptidoglycan. Biosynthesis of the peptidoglycan. Now, the process and amino acid sequence may vary according to the species. There are also various other points. Okay, for example, when we are trying to study how antibiotics kills the bacteria, as you all know, there are various antibiotics which can inhibit peptidoglycan biosynthesis. Okay, in this diagram you can see there are two examples given. One of them is vancomycin, the another one is an bacitracin. So vancomycin blocks step number 6, where NAG pentapeptide is attached. Okay, this will be blocked by the vancomycin, and the bacitracin, okay, which blocks the transfer of bactopenol to the another side of the membrane. Okay, another side of the membrane. Now moving further with the transpeptidation, you can clearly see the differences between E. coli and Staphylococcus aureus peptidoglycan. Okay, now in the case of E. coli with direct cross linking, the typical of gram negative bacteria. Okay, this one is a cross linking. Okay, and you can see the systems here. Okay, in the case of Staphylococcus, which is a gram positive bacterium, NAM is, as you all know, N-acetyl glucosamine. Okay, polysaccharide chains are drawn opposite to each other for the sake of the clarity, and two chains lying side by side may be linked together. Okay, now you can see where it gets attached. In the case of Staphylococcus, okay, they get attached with the lysine on one side, and another side will be aligned, whereas it differs in the case of E. coli, whereas the process differs in the case of E. coli. Okay, so what is the role of cytoskeleton in the cell wall synthesis? Okay, there are also various enzymes, okay, which you will study more in your master degree. Okay, so I'm not going to discuss about the enzyme, about the genes and more enzymes. The cytoskeletal structures formed by the MRE-BCD proteins control the cell shape by controlling the cell wall synthesis. Okay, so these proteins MRE-BCD basically helps in controlling the shape, okay, of the cell by controlling the cell wall synthesis. If there are any mutations okay, if there are any mutations which lead to defects in the bacterial cell, are directly associated with the defect in the cell wall synthesis. Okay, so if there is any problem in the shape or whatever the problems, or any due to the mutation, or it may be spontaneous, or it may be even induced mutation, whatever it may be, okay, due to one or the another reason, the cell wall has some different shape than regular, it can be due to the defect in the cell wall synthesis. And multiple mutations in PBP genes can convert rod-shaped E. coli and Bacillus into round or branched cells. Okay, so a multiple mutations in this gene, okay, PBP genes can convert rod shape and Bacillus subtilis, E. coli and Bacillus subtilis into round or even branched cells. Similarly, another mutation in TAGF gene, which is basically involved in teichoic acid, okay, which converts Bacillus subtilis into round cells, as I hope you remember where teichoic acid is present in which type of bacteria. Okay, now patterns, you must have already studied these patterns of cell wall synthesis during your semester 1, okay, where how cell divides, okay, cell wall synthesis patterns of new cell wall synthesis in growing and dividing bacteria, the streptococci and some other bacteria rod shape, okay, the zones of growth in turquoise, and the actual situation is more complex than indicated because cells can begin divide again before the first division is completed. So just an introductory type of pattern of the cell wall synthesis. Okay, now you can clearly see here the septal region, okay, the cell, it starts to divide, okay, further divides, and finally two different types of cells will be formed, where here the new cell wall you can clearly see, okay, the new cell wall here you can clearly see, okay, this is how cell wall synthesis works. Okay, now there was a problem while playing a video. Okay, I'm going to play the video again.
Most bacterial cell walls contain peptidoglycan. This structure is essential for maintaining the structural integrity of the cell. Peptidoglycan is a structure that is unique to bacterial cells. This combination of essential and unique makes peptidoglycan an excellent target for antibiotics. The precursor subunit of peptidoglycan, UDP-N-acetylmuramic acid, is synthesized in the cytoplasm. A lactyl group is added to carbon number 3 of UDP-N-acetylglucosamine to form UDP-N-acetylmuramic acid. 5 amino acids are added sequentially to UDP-NAM to form a pentapeptide side chain. Special adding enzymes are used to add these amino acids so that tRNA and ribosomes are not involved. In the case of Escherichia coli, L-alanine is added first, then D-glutamic acid, then mesodiaminopamelic acid. Finally, the dipeptide, D-alanyl D-alanine, is added to form the pentapeptide side chain. The NAM pentapeptide is transferred from UDP to bactoprenol phosphate at the membrane surface. Then NAG is transferred from UDP-NAG to the bactoprenol NAM pentapeptide to form the final disaccharide pentapeptide precursor. The completed NAM-NAG precursor is transported across the membrane to its outer surface by the bactoprenol carrier lipid. The disaccharide pentapeptide is then transferred to the growing end of the peptidoglycan chain to lengthen it by 1 repeat unit. As the bactoprenol pyrophosphate returns to the inside of the cell membrane, one phosphate is removed by a pyrophosphatase, leaving a bactoprenol phosphate to accept another NAM pentapeptide. Finally, the peptide side chains are linked together by transpeptidation reactions, forming peptide bonds. In E. coli, the free amino group of diaminopomelic acid is linked to the carboxyl group of the subterminal dealanine, releasing the terminal dealanine. Other enzymes remove the terminal dealanine from side chains that are not cross-linked.
Okay. Everyone, please join the lecture of WebEx of 8:40. Okay. Please join the lecture of WebEx of 8:40. And please comment present sir for your attendance.